Hi everyone, I hope you all are doing well. So my name is Harman Sharma and me and my partner named Jamuna, we both are working on the term project named as Evaluation of Inhibitory Effect of Terminal Chebula's Ethanol Extract on Cryogenic Bacterium named as Streptococcus mutans. So let's move towards the introduction of this topic. Okay, so it's basically clear from the title of our project that we are working on the cryogenic bacteria, and for this cryogenic bacteria we are basically use Streptococcus mutans because Streptococcus mutans is a highly gram positive bacteria which has all the characteristics which we require as a cryogenic bacteria to see its effect, to destroy it and to further proceed it in our experimental research. Okay, so in order to take a deep look on this cryogenic bacteria, so what's the purpose of this bacteria? What's the name of this? What's the meaning of this term, right? So cryogenic bacteria basically focuses on the human dental activities. They basically attack the human's teeth, okay, and they try to destruct them. They try to destroy them. In scientific term, cryogenic bacteria live in biofilm, and these biofilm bacteria basically attack on the surface of the teeth. It metabolizes sugar from teeth and further produce acids, and these acids further results in the erosion of teeth and results in the tooth decay and cause of various cavities. As we all know that the biofilms are insoluble. Similarly, these biofilms produced by the Streptococcus mutans, the cryogenic bacteria, are insoluble and they exhibit their resistance against the antimicrobial substances. Okay, and oral hygiene is necessary to control the proliferation of such infection causing bacteria and to prevent the dental caries. If we take any antimicrobial compound, any antibiotic from outside to get rid of these type of bacteria, similarly these cryogenic bacteria show their resistance. They start fighting with them and try to show their growth. They try to continue their growth because of their resistant behavior. Thus, as the need for new treatments and ingredients is increasing, finding natural compounds that can inhibit the growth of cryogenic bacteria plays a significant role of interest. So basically this is our results, this is our research that basically focus upon that we are trying to finding a natural compound, a natural antibiotic which help us to get rid of these type of cryogenic bacteria which proves very harmful for our human body, for our teeth. Okay, so in our project, we use terminal chebula as an antibiotic towards Streptococcus mutans, a gram-positive cryogenic bacteria. So in the lab, when we were performing our experiment, everyone was like shocked that what is this? What is this hard seed look like? Like what we call this? Okay, and where you get it from? What's the purpose of this seed? This is the hardest one. How we gonna use it, right? Okay, so let's take a look on this. So basically, terminal chebula, we use it as an antibiotic. Yeah, it looks hard. It looks like a seed because terminal chebula is a tree that belongs to the family Combractaceae and usually grows in the tropical and subtropical regions. Terminal chebula is already known for its use in various traditional medicinal applications because of its antibiotic nature, and previous studies shows that the terminal chebula fruit indicated that it contains phytochemicals such as the sapogenesis, anthraquinone derivatives, tannins, and flavonoids. Among all these chemicals, tannin is one of the main compounds which is responsible for the efficiency of this fruit. So terminal chebula contains tannins ranging from 32% to 45%, which includes gallic acid, chebulic acid, ellagic acid, chabulinous acid, and tannic acid. Clinical trials involving terminal chebula fruit extract as a mouthwash have shown reduction in build-up and juvenile inflammation of Streptococcus mutans. So in short, we use terminal chebula, which is a fruit we taken it out from the tree of terminal chebula. It is a dry fruit. It is a kind of dried fruit. We use this fruit, we use the powder form of this fruit in order to test the antimicrobial activity of this fruit towards the Streptococcus mutans, which is the cardiogenic bacteria responsible for the diseases caused in the teeth or the cavities, the tooth decay caused in our teeth, human teeth. We're gonna work on this terminal chebula to check whether it proves the positive antibiotic or not. Okay, so according to the scientific report, let's take a look at the objective of our report. Okay, so our objective is to evaluate the antimicrobial efficiency of terminal chebula ethanol extract against Streptococcus mutans, which we already discussed above as well, right? And what's the hypothesis? What we think it will be? So according to our thinking, the proliferation of Streptococcus mutans, a common oral bacterium responsible for dental caries, will be suppressed by ethanol extract of terminal chebula. So our thinking, according to our thinking and according to the scientific research as we did, we think that yes, the Streptococcus mutans will be suppressed by terminal chebula, will be suppressed by its antimicrobial activities. So let's see what's going on. We will perform the experiment and let's see what's the results and what we get out of these results. So in order to perform the experiment in a well-organized manner, we make a list of methods used for this experiment, which include firstly the extraction of terminal chebula with the help of ethanol. Secondly, the isolated pure colonies from the master plate of Streptococcus mutans. Thirdly, it's gonna be the viable cell count. Fourth, drug susceptibility test. Fifth, minimum inhibitory concentration. And the last one, minimum bactericidal concentration test, which we also call MBC. So the first and foremost step for our project was ethanol extraction of terminal chebula. As we see on the slide, it looks very interesting, right? And yeah, it was very interesting when we performed it as well. So for the first step, we used the fruit, the hardest dried fruit of terminal chebula. We take a lot of fruits and we put it into the grinder to grind it and get it converted into the powder form. So after getting the powdered form, we take 50 gram of that powder and we put it inside the Erlenmeyer flask, and by adding 200 ml of ethanol into the same flask, we put it inside the shaker, where we ensure the complete and continuous mixing of ethanol with terminal chebula for 48 hours. After the completion of 48 hours, when we take that solution out, we perform the vacuum filtration in order to filtrate our solution well and to get the purified form with the help of the filtration apparatus and the suction pipe. With the help of a filter paper, Wortmann filter paper, we put it on the top of the cuvette, and when we pour the solution having ethanol and terminal chebula inside, when we pour it on the top of the filter paper, all the solid particles collected on the top, whereas all the liquid form of terminal chebula and ethanol goes down into the flask. Then we use that liquid form as a purified solution of ethanol extract and we put it inside the fume hood for evaporation. After 48 to 72 hours, all the ethanol got evaporated and the powder form of the solution left behind, which we sucrashed and collected inside the tube by adding 1 ml of ethanol again and make it a broth to use for the further solution, for the further procedure. So basically in this step, in this first step of our project, we extract out all the nutrients, all the supplementary particles from terminal chebula, which are responsible for its antibiotic nature, okay, with the help of ethanol. And this process is known as to be the ethanol extraction of terminal chebula. So now we got the ethanol extract, we got the extract of terminal chebula in the broth form, which we're gonna use for the further steps to complete our experiment. Okay, so secondly, we perform the streaking to get the isolated colonies. For this step, we take the culture, Streptococcus mutant culture from the master plate. There were the colonies, and to ensure the pure culture, we took one colony and streak it aseptically on the BHI agar medium and incubate it for 35 degrees Celsius for 48 hours to get the purified colonies formed for our experiment. Then the next step was the viable plate count. In the viable plate count, we take 3 different concentration of EETC. EETC, that is the ethanol extract of terminal chebula, having the concentration of 2.5 milligram per mils, 5 milligram per mils and 7.5 milligram per mils. So we performed the separate plate technique in 3 triplets. We performed the spread plates in triplets and we added the Streptococcus mutant on the each plate having different concentration of EETC to check whether the EETC is working or not, whether the EETC shows its antimicrobial properties against the Streptococcus mutants or not, and at which concentration does it affect more or at which concentration it affect less. So we used 3 different concentrations and we try to find out the effect of EETC at different concentration levels against the growth of Streptococcus mutants with the help of spread plate technique. So in this technique, in this method as well, we use the incubation temperature of 35 degrees Celsius for 48 hours to check whether the Streptococcus mutant cells fight with the antibiotic that we added, EETC, and how much they able to grow their cells themselves or not. Thus, after performing the viable cell count, we perform the disc diffusion method. In this method, we take the broth culture of Streptococcus mutants and with the help of swab, we try to make a lawn of culture, a huge lawn of culture on the BHI agar plate. Alternatively, we take some discs of Whatman filter paper and we try to dip these discs in the different concentrations of EETC. The 3 concentrations that we were used for our experiment was 25 milligram per mils, 50 milligram per mils and 100 milligrams per mils. By dipping each disc in each different concentration of EETC and putting it on the top of the BHI agar having the lawn of Streptococcus mutants, we incubate it for 35 degrees Celsius for 48 hours in order to check the growth of the Streptococcus mutants, in order to check the susceptibility or resistance of Streptococcus mutants against the EETC, that is the antibiotic that we added, the ethanol extraction of terminal chebula. So basically in disc diffusion method, as we all know, after getting the result, we use the vernier scale to measure the zone of diameter and we can check that whether our bacteria is susceptible or resistant towards the antibiotic that we have added. So this is gonna be further discussed in detail in the results section. So the next step performed was the minimum inhibitory concentration test, that we also known as to be the MIC test. So as we all know that the minimum inhibitory concentration is a key parameter in antimicrobial susceptibility testing, providing the lowest concentration of antimicrobial agent that inhibits bacterial growth. In this study, the MIC of terminal chebula ethanol extract against Streptococcus mutans was determined using a broth dilution method. And in order to perform this method, we took 12 sterilized screw cap tubes, and out of the 12 sterilized screw cap tubes, we used one tube for the double standard 2 ml of double standard BHI broth, whereas the rest of the 11 tubes we put the single standard BHI broth. And in order to add the varying amount of EETC, the antimicrobial compound, the EETC inside these tubes, we start adding 2 ml of EETC from the beginning into the double standard tube, and from that double standard tube we start doing the dilutions. And in the form of dilutions, we took 2 ml of each 2 ml out from each tube, from the first tube till the last 12th tube, to make sure there will to make sure the 2 ml of same amount, same volume in each tube. At the end, the rest of the 2 ml that we take out from the 12th tube gonna be discarded into the waste disposal. After adding the varying amount of EETC in the broth culture of BHI, we started adding 2 drops of Streptococcus mutans culture into each tube, and we incubated it at 35 degrees Celsius for 24 to 48 hours. This step in the experiment was conducted in duplicate to ensure the accuracy. The MIC determination provides crucial information on the potency of terminal chebula ethanol extract against Streptococcus mutans, helps in understanding of its antimicrobial effectiveness. And by means of this test, we ensured the MIC, which was the lowest concentration of the extract that prevent the visible bacterial growth, as observed by the means of turbidity, which gonna be further discussed in the results section. Okay, last but not the least, in order to protect our teeth from the bacteria, from the cardiogenic bacteria, Streptococcus mutant, we perform one more test that is known as to be the MBC, minimum bactericidal concentration. Okay, scientifically, the MBC was determined subsequently using the broth dilutions from MIC test. After identifying the MIC, samples with concentrations equal to or higher than MIC were streaked on the brain-heart infusion, that is a BHI agar plate, by dividing it into the compartments. Okay, so the MBC was then defined as the lowest concentration at which no viable bacterial colonies were observed after incubation at 37 degrees Celsius for 48 hours. This step helped elucidate whether the extract not only inhibits the bacterial growth but also effectively kills the bacteria. The experiment was conducted in duplicates, resulting valuable insight into the bactericidal potential of EETC, contributing to a comprehensive understanding of its antimicrobial properties against Streptococcus mutans. However, the pictures describing the results of the MBC will be described in the results section.
Hello everyone, this is Jamuna Bhupati. Now I'll unfold the results we got from this experiment. So like Harman mentioned before, the test organism in this experiment is Streptococcus mutans. At first we isolated pure colonies of Streptococcus mutans on brain-heart infusion agar. As we needed the culture in liquid media to conduct the test, we inoculated this pure culture into BHI broth. After incubation, we observed growth in BHI broth through its turbidity. Then we wanted to estimate the concentration of this culture, so we followed spread plate technique and we found that the concentration was 1.1 times 10 to the power 9 CFU per ml. Now coming to the main results, the first test we conducted was to observe the inhibitory effect of ETC on the growth of Streptococcus mutans. So here we treated the culture with 3 different concentrations of ETC. Here you can see 2.5, 5 and 7.5 milligram per ml concentrations were used. This treated culture was diluted and spreaded on agar plates to count the viable cells. So after incubation, we found a decreasing trend in the result. When the concentration of ETC increased, the number of colonies on the plates decreased. So this means that Streptococcus mutans growth was suppressed to some level by the effect of ETC. Following the inhibitory effect test, we wanted to determine the minimum inhibitory and bactericidal concentrations of ETC. So we followed two-fold broth dilution method. After two-fold dilutions, the concentrations of ETC in this test tubes range from 50 milligram per ml to 20 microgram per ml. So after incubation, there was no growth in the first two test tubes, that is test tube containing 50 milligram per ml and 25 milligram per ml concentrations of ETC. Hence, the minimum concentration that was effective at inhibiting S mutants was 25 milligram per ml. Also, in the MBC test results, we observed no growth on streaks from 1/4 and 1/2 dilutions. So the minimum bactericidal concentration was also found to be 25 milligram per ml. So both MIC and MBC was similar. Now the final test was to observe the susceptibility of Streptococcus mutans to ETC through the disc diffusion method. Here we observed clear inhibition zones around all the 3 discs infused with 25, 50 and 100 milligram per ml concentrations of ETC. The size of inhibition zone was linear to the concentration of ETC infused to the disc. We can see clearly in the next slide. The diameter of inhibition zone around 100 milligram per ml was larger and small around 25 milligram per ml concentration of ETC. So this clearly explains that S mutants is susceptible to ETC. So finally, all these test results supported our hypothesis, which we stated earlier, that proliferation of Streptococcus mutans will be suppressed by ethanol extract of Terminalia chebula. It was evident from the inhibitory effect test that S mutants growth was suppressed by ETC, and the organism's susceptibility was also confirmed from the results of disc diffusion test. Since both MIC and MBC values we found in this experiment was 25 milligram per ml, maybe further dilutions in between 25 to 13 milligram per ml could help to find the accurate minimum inhibitory concentration, since 25 milligram per ml was able to kill the bacteria as we observed in MBC test. So to conclude, all these findings help to verify the antibacterial effect of ETC against S mutants, and so we can say Terminalia chebula can be a good natural ingredient to incorporate into oral care products for the prevention of dental infection that are usually caused by S mutants. More testing on the susceptibility of other oral bacteria like lactobacillus and Streptococcus sobrinus to this ETC could help to further evaluate its potential in controlling dental caries. So that's it. Thank you everyone for your attention. We would like to express our sincere thanks to our professor for the guidance and feedback, and we would also like to extend our thanks to Biotechnology Department for all the resources they have provided for this project. Thank you so much.
Thank you so much for watching our presentation. I hope you enjoy it a lot. Have a nice day. Thank you so much, everyone.